Okay, so hello, my name is Jeff Smith, and I'm the Strategic Partnership and Outreach Specialist for the Institute of Cannabis Research, hosted at Colorado State University, Pueblo. Welcome to the Cannabis Research webinar series, which is presented by the Institute of Cannabis Research and the Lambert Center for the Study of Medicinal Cannabis and Hemp at Thomas Jefferson University. This webinar series, in collaboration with the Lambert Center, is one of the many ways that the ICR works to promote the dissemination of cannabis research. For example, please see our website, which can be found by Googling the words Institute of Cannabis Research. From there, you can navigate to our Outreach tab and find the current e-newsletter. Also, you can navigate to the Research tab in the website to view summaries of each of the research projects that the ICR is currently funding. Stay tuned, because soon we will have a new website which will feature an additional 6 projects which we are initiating this year. All of our research is done by the best, brightest researchers that the state of Colorado and their collaborators has to offer at our best institutions. This is going to bring our research funding to a total of 21 projects this year. Also, please note the link to the Journal of Cannabis Research, which the ICR sponsors. Before we get started with the presentation, I'd like to mention some logistical items for the webinar. Today's presentation will be followed by a short question and answer period. To ask a question, please do so using the Q&A feature in the Zoom toolbar. You can enter questions at any time during the presentations. We will address as many questions as time allows until the end of the hour. Please reserve the chat function in Zoom for technical issues. If you have problems that you need to ask about, ask them in the chat function. Both the Q&A and chat functions are on your Zoom toolbar on your Zoom screen. It's now my pleasure to introduce my colleague, Ruth Charbonneau, the Associate Director of the Lambert Center, who will be introducing today's presenter and moderating the webinar.
Thank you. So we are pleased to have Dr. Ryan Vandrey with us today. He is an experimental psychologist with degrees from the University of Delaware and University of Vermont. He is currently a professor at Johns Hopkins University Behavioral Pharmacology Research Unit. Dr. Vandrey's research focuses primarily on the impact of route of administration, dose, and chemical composition of cannabis products on resultant drug effects and pharmacokinetics. In addition, Dr. Vandrey has been involved with a broad range of studies related to the risks and benefits of medical cannabis use, the effects of cannabis use on sleep, cannabis withdrawal, and the treatment of cannabis use disorder, cannabis product testing, and developing measures of cannabis use behavior. We are pleased today to have him to discuss experimental analysis of acute cannabis and cannabinoid effects in healthy adults. And with that, I'll turn it over to you, Dr. Vandrey.
Thanks, Ruth. I appreciate it. And thanks for the invitation to come and share our science. So I'm not going to belabor too much here, but I want to kind of get going. I'll acknowledge disclosures that I have been a paid consultant or advisory board member for several pharmaceutical companies. So I want to begin the talk by just getting us all on the same page in terms of what we're talking about. I think it's more important now than ever to be careful with our terminology when we're describing cannabis and cannabinoid research. So cannabis is a plant that's made up of over 100 phytocannabinoids, which are chemical constituents that are unique to the cannabis plant, as well as hundreds of other terpenoids and flavonoids, which are not unique to the cannabis plant, but can have an impact on the pharmacology of cannabis, whole plant cannabis products. There are also a number of synthetic cannabinoids, and both phytocannabinoids and synthetic cannabinoids have a diverse pharmacology, so they don't all behave the same way. The primary cannabinoids, phytocannabinoids that we're interested in are CBD and THC, but there are a number of what we call minor cannabinoids and acid versions of these things that we're interested in. And the more science that comes around, the more research we do, we're finding that terpenes and flavonoids are also very important to potential therapeutic and potentially harmful effects of cannabis as well. So when we're talking about cannabis and cannabinoid research, it's important that we recognize this diversity of pharmacology, the complexity of the chemical constituents of these products, and we refer to things by individual chemotypes because the cannabis term, broadly speaking, is too diverse now. So when we're communicating with each other, when we're communicating to the public, we want to identify the dominant cannabinoid that we're referring to in a particular product. So a high THC, high CBD, balanced THC, CBD product. We want to identify the route of administration that's intended for a product to be used, whether it be inhaled, oral, topical, or some other form. And in terms of labeling, I think as a field want to start to operationalize terms like isolate, full spectrum, and things like that. And as much as we can to use this consistently and not just use cannabis in our terminology and our communications. So where my research comes from is really the identification of this gap between science and retail cannabis products. There's just an incredible diversity of products, formulations, and intended routes of administration on the market today. And we don't have a lot of science to understand the nuance between these different products. Understanding how and when it's appropriate to use cannabis or individual cannabinoids for therapeutic purposes is lacking based on this diversity. We can't necessarily generalize across all products. If we see an RCT for one, that it means it's going to work for all of the other stuff. And we really need to work on improving education and communication across this stuff. So where I try to focus my efforts is really trying to take what is out there, what is known, and filling those gaps to try to minimize the likelihood of overstating safety and efficacy and understating risks. So as in science, it's a team approach. We have kind of dubbed ourselves the Cannabis Science Laboratory at Johns Hopkins. There are a number of folks here, in addition to a number of folks not listed here, that help me do this research. We also have a number of outside collaborators that I'm not going to get into now, but I'll identify and acknowledge at the end of the slide. But our laboratory is building out, and we've got a little QR code. If you scan that, you can go to our website and see the kinds of active projects that we have going on, some of our publications and things like that. We're also looking for postdoctoral fellows. So if anybody's interested or knows somebody who might be interested in that, please go through our website or email me later and let me know. So what we're trying to, what we've been working on and where we're really focusing is really a systematic evaluation of cannabis products with understanding the impact of variations in the route, dose, formulation, and chemotype of the products and how those things impact acute drug effects. We're very interested in drug interactions, research methods development, detection of impairment, and the health impact of medicinal cannabis use. So what I'm going to focus on for the most part today is our kind of phase 1 PK/PD type protocol, where we have this root protocol design that we use across most of our experimental tests. And it's the basis for most of what we do. So we take the same kind of root protocol and we modify it a little bit to study those variations in products that I mentioned previously. But by and large, what we do in these studies is we recruit healthy adults from the community, always try to get a good balance of males and females. Most cases, we have people required to have used cannabis previously, but typically not for at least 30 days. So we're trying to minimize the effect of tolerance in our experimental tests. On occasion, we allow some infrequent past month use. It depends on the study and how frequently we're trying to run sessions and the doses we're trying to evaluate. In most cases, we use a within subjects crossover design to minimize individual level variance in terms of determining differences in drug effects across doses, routes, or other formulation manipulations. We tend to formulate cannabis products in-house with our pharmacist with input from the industry. This allows us to obtain products manufactured in a GMP formula method in terms of synthetic pure compounds. And then we will have the capability of using the exact same source cannabis or cannabinoid product, whether it be pure THC, pure CBD, a high THC distillate, a high CBD extract, but then we can use that source product to put into an edible or a vapor cartridge or make different types of edibles. So we can look for these formulations and not have individual differences in the source cannabis material. In each study, we do controlled dosing under observation with medical supervision, and we will evaluate our participants pre-dosing at baseline and then multiple times after dosing. And typically that involves evaluation of both pharmacokinetic and pharmacodynamic assessments. And we usually will require at least a 1-week washout in between doses. I also want to note that all of our studies are conducted under INDs with the FDA and under my DEA Schedule I researcher license. I think that that's important to have that kind of federal oversight of what we're doing. And all of our research products are fully federally compliant. Now, the assessments that we do, as I mentioned, accomplish both pharmacokinetic and pharmacodynamics. Hopefully everyone can see my pointer here. So we collect blood, saliva, and urine from participants throughout these test sessions each day. We have people use a visual analog scale to tell us subjectively how they're feeling, whether they feel a drug effect, anxious, nervous, tired, sleepy, impaired. And then we have a battery of cognitive performance tests that we utilize. We have a working memory task at the bottom left corner. You see the paste serial addition task. Bottom right, you see a divided attention task where people have to monitor numbers in the corner of the screen and click the mouse button each time the target number in the bottom middle matches the one in one of the corners. Meanwhile, this box bounces back and forth, and you have to keep that little plus symbol on top of the box as much as you can. We have a psychomotor test, the digit symbol substitution task, where you have to repeatedly type patterns over and over on using the computer keyboard. And then we have an iPad-based app called the Druid app that we've been utilizing that kind of pulls a lot of these different cognitive domains into a single 2-minute task that also adds balance. And we collect blood pressure and heart rate using an automated monitor. So what I want to do is over the next, 30 minutes or so is just to kind of walk through a series of experiments that kind of tells the story of what we've been doing and what we've been finding in some of our research. So we started out with all of this looking at some basic kind of doses, placebo, what we called at the time a low dose, a 10 milligram dose of THC. This is high THC cannabis, whole plant botanical cannabis at this point in time, and a higher dose of 25 milligrams of THC. So we took the same plant material and we had people smoke it, vaporize it, or orally ingest it in a chocolate brownie. And what we found was that route of administration and method of administration impacted both the time course and the magnitude of drug effects seen. So this is ratings of subjective drug effects at the 10 milligram and the 25 milligram dose. I'm not showing placebo because it was 0 across the board. We didn't have much in the way of of placebo effects with the oral or vaporized doses. With smoked, you get some interoceptive effects as a little bit, but it's not terribly interesting. So when we look at these dose effects, you see dose orderly increases going from 10 milligrams to 25 milligrams in terms of subjective drug effect. We see inhalation with vaporized and smoked giving you immediate peak drug effects with a fairly steady decline back to baseline values, a slower onset and a longer peak duration of drug effect with oral dosing. The other thing that's fascinating here is that the peak drug effects between oral dosing and smoked dosing are the same. They just occur at different times, and that vaporization gives you the highest magnitude of drug effect. And this was observed for subjective drug effects, not just feeling any drug effect, but all of the other kind of minutia there in terms of feeling sedated, feeling that hungry or having the munchies, feeling impaired, showed the same pattern of effects. But a key difference between this 10 milligram and 25 milligram dose is a 10 milligram dose, which is the maximum unit dose for a high THC cannabis product in most states that allow retail sale. It was really well tolerated by most people taking it. But when we bumped it up to about 25 milligrams, and we actually went to 50 milligrams with our oral dose as well, that's when people started getting uncomfortable. And one of the most common adverse effects that we saw was people reporting feeling anxious or nervous. So when we identified that, we identified that dose of 25 milligrams as inducing anxiety in a handful of people. We started talking with our my friend and collaborator, Ethan Russo, said, all right, well, what can we do about that? What is something that we see the industry claim? And what we see is people saying that the terpene limonene can actually provide stress relief and reduce anxiety. And Ethan wrote a really nice review back in 2011 that identified synergistic interactions between individual cannabinoids and terpenes, and specifically hypothesized that limonene could mitigate the anxiogenic effects of high doses of THC. Now that sat for a little while, and then we were able to get a grant to study it, and we were able to put empirical data behind this hypothesis. And what we found is when we went into an experimental laboratory environment and gave no THC through vaporization, we found no drug effect. When we gave people a little bit of limonene, it really didn't produce any anxiety. We gave people 15 milligrams of THC, we induced a little bit of anxiety. When we upped it to 30 milligrams, we induced more anxiety. But then as we increased pure THC through vaporization, and we added increasing doses of limonene, we saw a dose orderly reduction in the self-reported ratings of anxiety. So we're seeing here that limonene can reduce the anxiogenic effects of high doses of THC. This was also seen for self-reported ratings of paranoia, but it didn't change overall ratings of total drug effect. In fact, it was very specific to this anxiety-induced effect. It didn't alter cognitive performance on any outcomes or any other subjective drug effects like getting the munchies or things like that. So this was really interesting and is something that we can utilize and identify and try to push forward in drug development to make THC containing medications more tolerable to individuals. So one of the other key findings that we see, again, at 10 milligrams, we see impairment of working memory. And so 10 milligrams is really no different across different routes of administration at 10 milligrams, no significant change from placebo, no significant change from baseline either. But what we see is when we bump up to a 25 milligram THC dose, we see substantial impairment of performance on this memory task. And this is a very simple memory task, adding 2 + 4 equals 6, 4 + 3 equals 7 in a sequential and somewhat rapid manner. And the ability to do that becomes significantly impaired at 25 milligrams of THC. So one of the other hypotheses that Dr. Russo had was that alpha-pinene could mitigate some of the working memory impairing effects of high doses of THC. So we're right in the middle of running an experiment that had really the same kind of approach as the limonene study. And what we're looking at here is working memory performance on that serial addition task. And this is the total number of correct change from baseline. When they get placebo, they get a little bit better over time with the task. When they get just pinene by itself, it doesn't really impact performance much. But when they get 30 milligrams of delta-9 THC, performance is impaired. When we add increasing doses of alpha-pinene, performance recovers. Now, the 15 milligrams isn't really continuing improvement beyond 5 milligrams, and we're getting an inverted U-shaped function, which is common in behavioral pharmacology. I'll note that we only have 8 participants through the study at this point, but it'll be interesting to see if this plays out and we get statistically significant effects there. But it's something that we're going to continue to investigate. In addition to that, we see impairment of memory encoding on a shopping task memory encoding and consolidation assessment. We see impairment when we give THC regardless of the alpha-pinene dose, not really much, a little bit of a signal of recovery with 5 milligrams, but not much. But retrieval is actually showing at that 5 milligram pinene dose is kind of the magic dose right now. Full recovery of impairment at that dose and looking similar to just pinene by itself. So stay tuned for that. And then similar to the pinene, we really saw no impact on overall drug effect ratings or really anything else with other outcomes. So we're seeing signals of individual terpenes and modulating the effect of THC, which I think is valuable for differentiating entourage effects and full spectrum versus isolated pure cannabinoids to be used therapeutically. Another key area that I think we need a lot more science to bear and to inform regulation and policy is related to driving and workplace safety. So as cannabis becomes legalized, we're faced with the challenge of what do we do? How do we police this? How do we prevent airplane pilots and forklift drivers and school bus drivers from driving under the influence, but allowing them the freedom if they live in an area where it's been legalized for either therapeutic or non-therapeutic uses to engage in that freedom that other people are allowed to do. So there's an immense interest in identifying something that can be used to detect impairment and to prevent impairment in workplace or driving settings. So we've been doing a little bit of work on that, and I'm going to go back to our whole plant administered through different routes of administration to highlight one of the key challenges in all of this. So with alcohol being legalized, we have a very easy to obtain, very rapid indicator of whether or not somebody's been drinking alcohol and is likely to be impaired at a given time. With cannabis, we don't have the equivalent of a THC breathalyzer that's very good or sensitive or has been demonstrated to be valid at this point. What most people rely on are THC levels in blood. And what I'm showing you here in this 2-panel graph on the left-hand side is blood THC levels after our 25 milligram THC dose in a high THC flower product across 3 different routes of administration. And on the right-hand side, we see subjective drug effects. So you can see that while we have this comparability in terms of pharmacodynamic outcomes between smoked and orally ingested and a little bit higher magnitude with vaporized, we see very different patterns with the blood THC concentrations. So we still see the immediate peak with inhaled products, but it returns down to 0 or near 0 levels before the subjective drug effect dissipates. And then, although we have comparable levels of pharmacodynamic drug effect in terms of subjective drug effect ratings, as well as impairment of cognitive performance, between oral and inhaled dosing, we don't see the same concentrations in blood THC levels. And the same thing goes for 11-hydroxy metabolites. So we see much lower concentrations. So establishing a per se blood THC level that would relate to impairment is greatly challenged by the fact that cannabis is consumed in different forms and different routes of administration have different rates of absorption, but not very different rates of magnitudes of pharmacodynamic effects. So per se limits are very challenging. And then to highlight that even further, I'll point out on this slide, this is data from a different study where we had people orally ingest THC doses, again, 0, 10, or 25 milligrams of THC, or vaporize 0, 5, or 20 milligrams of THC. And we looked at blood as well as oral fluid or saliva specimens. A lot of countries have adopted oral fluid testing for roadside. And we see, again, real challenges in trying to identify good thresholds. So in blood, as I showed you on the prior slide, we have this time course problem for blood in that it comes back to 0 within an hour, but the drug effects persist. Low concentrations with oral dosing. And on the oral fluid or saliva testing level, we have really high concentrations immediately and then back to 0 within 2 hours. So again, a disconnect in terms of the timing. And with oral dosing, we know our peak drug effects occur about 3 hours, between 2 and 3 hours post-dosing. And at that point, oral fluid concentrations of THC are down to near 0 again. So we have yet to identify a biomarker in blood, saliva, or urine that reliably predicts levels of impairment in THC in blood or saliva or urine. So we turned to field sobriety testing. So we did the study again where people are dosed orally or through vaporization of high THC cannabis at THC doses, delta-9 THC doses of 0, 10, or 25 with oral, 0, 5, or 20 with vaporization. And without drilling down into what each of these bars mean, what I'm showing you is 3 different commonly used roadside field sobriety tests. And you can see that the pattern in terms of the bars, the number of clues identified by trained drug recognition experts, really doesn't change with increasing doses, even though we see demonstrated impairment of cognitive functioning in our computer-based tasks in this experiment. So standard field sobriety testing is not a very good metric of whether or not somebody's impaired at the time. So what we did in that same study is we did our first evaluation of the Druid app. So this is, as I mentioned earlier, is a short iPad or iPhone or tablet-based, not to try to totally promote Apple right now, but a tablet or phone-based application that people can use to try to detect impairment. And the cool thing about this app is it's not necessarily specific to cannabis or alcohol or a benzodiazepine or sleep deprivation. It's really just looking for impairment of functioning, broadly speaking. And notice in this experiment, we slightly varied the doses of THC that we gave to people through the oral and vaporized routes. And that was intentional because those earlier slides that I showed you showed that vaporization produced a little bit stronger drug effects in terms of peak magnitude than the oral dosing did. And we picked the 10 and 5 milligram doses as our low dose and the 25 and 20 milligram doses here to identify the sensitivity of this to detect impairment and not necessarily just any drug effect. Because if you recall in our first study, 10 milligrams of THC given orally was really well tolerated, was not associated with cognitive impairment on our lengthy computerized battery of tasks, but 25 milligrams was. And so we wanted to test the sensitivity and specificity of this measure, and we actually found that it worked really well. So the lower doses did not differ from placebo but our higher doses did, and it followed the time course of subjective drug effect ratings. So we thought that the Druid app was actually really good. As I mentioned before, though, the main limitation of this here is it requires the establishment of some kind of baseline performance in order to interpret these changes that might happen as a function of exposure to cannabis or other drugs that could cause impairment. But this type of approach may be suitable for workplace scenarios where you need to assure that people are not impaired, but are allowed to, on the weekends, responsibly use cannabis for therapeutic or non-therapeutic purposes. So related to safety, and introducing our interest in drug interactions. My colleague Tori Spindle is currently running an experiment looking at cannabis alcohol interactions. And the reason for this is that there's extensive literature showing that alcohol impairs driving, and that you have an established 0.08% BAC or breath alcohol concentration that law enforcement uses, that workplaces can use to say, look, you've had too much to drink, you're likely impaired and you shouldn't be driving or working under safety sensitive environments. We also know from prior research that THC can impair performance and can impair driving. What we don't know very much is at what dose levels do the combination of these two things become important? And is it necessary to potentially adjust our BAC thresholds, or how do we look at THC to determine safety in these scenarios? And so in this experiment, Tori has looked at high THC cannabis, botanical cannabis products. This is an oral dosing study initially. Again, our 10 and 25 delta 9 THC doses, and we can see that simulated driving performance gets worse. So this is an impairment score. Higher doses mean worse driving. And so you see dose orderly effects. The higher the THC dose, the worse people drive. A 0.05 BAC, which is under the threshold in most US states, impairs people a little bit less than the THC does. But the 0.08, which is our threshold in most US states of you shouldn't be driving or shouldn't be at the workplace in a safety sensitive area, is actually a little bit above the THC by itself and is substantially higher than the 0.05 BAC. And interestingly enough, when we combine our THC with the 0.05, the low BAC, we get at a 25 milligram THC dose and a 0.05 BAC, the same level of impairment of driving as we do with a 0.08. So this is interesting because this person, if they're pulled over on the roadside and just given a breathalyzer, they've got a 0.05, they're not past the threshold, and they've orally ingested THC containing products. There's not really going to be, we know the field sobriety tests really don't catch at that too much. This person may get let go. And so this is something that we're going to have to try to figure out. Interestingly enough, subjectively, these participants across all dosing conditions except the low THC condition did not feel confident in their ability to drive. So we're going to need to rely on people self-policing to some degree. But we know that that's usually not very good. So that's an area that we really need to drill down on and to come up with really good objective assessments of impairment and potentially retool or rethink how we determine safety with regards to alcohol and cannabis exposure. The other area that I want to touch on now is looking at delta 8 THC, which has become pretty prevalent in the US. And how that compares to delta 9 THC. So delta 8 THC is proliferated in the US as a result of the 2018 Farm Bill, which descheduled hemp. And from hemp, you can take hemp-derived CBD, do a very quick chemical conversion and get delta 8 THC, which is very similar to delta 9 THC shown at the top. You just get your double bond over here shifted counterclockwise around the ring there. And what we end up with is a very similar pharmacology. So what we're doing right now is an experiment where we're giving people oral doses and vaporized doses of delta 8 THC of 10, 20, and 40 milligrams and comparing that to 20 milligrams of delta 9 THC. In each case, this is the pure cannabinoid. It's not a botanically derived. These are synthetically derived pure compounds. And with oral dosing, we see beautiful dose orderly increases in subjective drug effect. And we see that the delta 9 THC is about twice as potent as the delta 8 THC. But what we're seeing is as you increase the dose, you can get the same kinds of drug effects as you get with delta 9 THC. When we have it administered via vaporization, there's this subjective perception of a higher drug effect across all doses that really is not dose orderly. It's all about the same. There's really no difference across any of the active doses, which is interesting. When we look at impairment, and again, coming back to that Druid app, we see nice dose orderly effects. We see comparable magnitude impairment between our 40 milligrams of delta 8 and 20 milligrams of delta 9. That's similar to what we saw in subjective impairment. We're seeing more dose orderliness with our vaporization. And again, comparability between 40 of delta 8 and 20 of delta 9. Looking into the pharmacokinetics to try to explain this, if we're looking at oral dosing here, we're showing whole blood delta 8 THC concentrations on the left-hand side and whole blood delta 9 on the right-hand side. You can see the purity of our compounds. There's no delta 9 detected when we give delta 8. There's no conversion of one to the other. And the same thing for our delta 9. There's no delta 8 detected there. What we're seeing, though, is, the 20 milligrams of delta 8, the parent molecule is at the same concentration, but remember that the pharmacodynamics is that the 40 milligram, you need 40 milligrams of delta 8 to get the same drug effect as 20 milligrams of delta 9. So why is that? It's when we look at the 11-hydroxy metabolites that things are getting a little bit wonky. And so for those that aren't aware, the 11-hydroxy metabolite is psychoactive and has a little bit different pharmacokinetics, a little bit potentially better brain absorption, and possibly a little bit stronger affinity or potency at the CB1 receptor. And we see a lot higher concentrations, almost double the concentration of delta 9, 11-hydroxy delta 9 THC compared to the 11-hydroxy delta 8 THC. And more than double the inactive carboxy metabolite of delta 8 compared to delta 9. So we're seeing what looks like a more rapid and more complete metabolism of delta 8 THC compared to delta 9 THC, and that might be driving the potency difference in addition to affinity for the CB1 receptor. So with that, I think the take homes for the delta 8 story is that you can get the same types of drug effects from delta 8 as you can with delta 9. You just got to boost the dose. And the reason for that is metabolism and pharmacology. So I've talked a lot about delta 9 THC, delta 8 THC. I haven't talked much about CBD. So I'm going to take a moment on that. And we've done some studies where we give acute doses of CBD to different people in different formulations and different routes of administration. In a recently published study, we gave 100 milligrams of CBD vaporized and orally. This was pure synthetic CBD. And then we gave a high CBD flower that also contained 3.7 milligrams of THC, and we had them vaporize that. And we asked them about acute drug effects, and we really didn't see any acute drug effects, no difference from placebo with any oral dose formulation that we gave. When CBD alone was vaporized, we saw a little bit of a drug effect, but nothing anywhere near the same magnitude as when we added a little bit of THC to it. Now, when we looked at CBD by oral dose by different formulations, we looked at Epidiolex, CBD suspended in a pharmaceutical grade cherry syrup, and CBD encapsulated. We saw a very substantial formulation effect where there is the highest absorption with Epidiolex, which is an oil-based formulation. Next highest was the encapsulated formulation, and the lowest concentration was the syrup. And then we had people take the syrup after overnight fasting, and there was hardly any detectable CBD, only across 3 time points and at very low concentrations. So the same drug given in 3 different formulations, all by mouth, had very different absorption, and it was impacted by the formulation as well as gastric contents. And we also found in this study that CBD was not THC-like on any drug effect kind of specific outcomes. We saw no paranoia, no memory effects, no heart racing, no increase in heart rate, no subjective feeling of that. And the low dose of THC, 3.7 milligrams, really didn't drive much of a drug effect, a little bit of the dry mouth, a little bit of a subjective drug effect, but no impairment of any cognitive functioning with a 3.7 milligram vaporized THC dose. And we've recently replicated that same 3.7 milligram THC dose with an oral dose formulation and seen the same thing. That low dose of THC really doesn't produce much in the way of drug effects, definitely no impairment of cognitive functioning. So hemp products that contain low doses of THC, we're trying to figure out the threshold there, seem to be pretty safe in terms of impairment and intoxication. But the key is that they drive positive screenings on urine drug tests. So we've seen positive drug tests as low as 2 milligrams of THC. We're currently evaluating 1.5 milligram, but we don't have data on that just yet. So we'll be reporting that over the next 3 or 4 months. But while CBD by itself seems pretty inert, it can interact with THC substantially. So we had a paper that just came out recently showing that a 20 milligram dose of Delta-9 THC combined with a 640 milligram dose of CBD produced substantially higher drug effects than just a 25 milligram dose of THC. And that was driven by pharmacokinetic interactions. So CBD, we determined, was a substantial inhibitor of Delta-9 THC metabolism, shown in blood levels here. It doubled the concentration of Delta-9 THC in blood, increased the 11-hydroxy metabolite tenfold, and the result of that was a substantial increase in adverse effects and impairment of cognitive performance. I'm going to go a little bit faster now because we're running low on time. But in addition to interacting with Delta-9 THC, CBD also inhibited the metabolism of several CYP450 probe drugs. So what this means is that medications metabolized through CYP2C19, CYP3A, 1A2, and 2C9 are likely to be inhibited in terms of clearance when combined with CBD. Now, we gave a pretty high dose of CBD here, 640 milligrams, but the PK modeling that my collaborators did show that you're likely to see inhibition of metabolism of those same CYP enzymes at lower doses of CBD. So dose adjustments in drug combinations for therapeutic CBD are likely to be needed. We've are halfway through an acute CBG or cannabigerol study. Interestingly, CBG is looking more like CBD than anything else, and that acutely it doesn't produce much in the way of interoceptive drug effects, no change from placebo up to 200 milligrams, and no impairment of cognitive functioning. And I want to take just another minute here and highlight that most of what we do is doing these controlled laboratory experiments. We're interested in, and we're pushing to do more experiments where we're moving these lab methods into the field. And we recently completed a an outpatient kind of remote observation study where we looked at medical cannabis patients in Maryland that were kind of new to cannabis, had clinically significant anxiety and depression. And we engaged them in a study where they kind of took pictures of the products that they were using, told us about them, used them in real time, and gave us feedback on the experiences. And what we were able to do is look at different routes of administration and acute effects of people using these products in their home environment. And what we saw is people reported increases in their subjective feelings of getting high. They predominantly used high THC versus high CBD products. And that even though they felt high, they saw acutely, and these were assessments done immediately pre-dosing and then post-dosing at the right amount of time to kind of get to peak drug effect levels, acute reductions in anxiety. We also saw acute reductions in depression. And that the reductions in anxiety and depression were dose specific. So again, seeing inverted U-shaped functions, especially for depression, where 1 to 5 milligrams really wasn't high enough. 6 to 10 was kind of the sweet spot. But man, if you got up to 16 or higher dosing, you were actually not getting much in the way of reduction in anxiety, depression. And that's at an aggregate level. There were individual cases where you induced anxiety or depression with those higher doses. So it highlights the importance of instructing people on the right dose to use and tailoring doses to the individual. But then longitudinally in this experiment, we were able to show clinically meaningful reductions in both anxiety and depression over time. So this is showing reductions over 3 months after initiation of medicinal cannabis use from people who were not using any cannabis previously. So we've got a lot of exciting stuff coming up. We're going to do some oral limonene THC interactions, some THC myrcene stuff, more things on driving, looking at delta 8. We've got a real big longitudinal observational study employing some of those EMA and phone-based assessments that I just told you about. I'm going to skip the summary because it all kind of makes sense. It's just restating what I've said. But basically, it's important to know that we need to understand the nuance of all of these different formulations, types, chemicals, and methods of using cannabis, and how that can impact differences at the individual drug effect level. And I'd encourage anybody here who's not doing this kind of stuff to do it. Let me know if you need help navigating the complicated regulatory scene there. But we need more people doing this stuff because there's so much more to do. I need to acknowledge everybody that helps with all of this stuff. I've got a ton of people that I work with. It's super fun to do it. Lots of people contribute to this. And again, I want to thank everybody for your attention. Here's the QR code for our website, my contact information. And I think I left enough time for us to handle some questions here, but anything we don't get to, feel free to reach out at another time. So now I'm going to shut up and take it to the questions.
Thank you. Thanks for providing your email address as well if we don't get to all the questions. So the first is part of your early part of your presentation it's 2 parts. Was the limonene co-administered with the THC? And did you look at the ability to have any rescue properties by administering the limonene later after first reporting the feeling of anxiety or nervousness?
Yeah, so the drugs were administered simultaneously using a vaporizer. So we had an individual dose pad, and both drugs were given concurrently and inhaled at the same time. We did not study any rescue effects. So if you give limonene later on, can it help? We'll have to figure that out. My guess is probably, but that's something that still needs to be determined.
For the Druid, there seems to be a wide variability at baseline. How do the individual data look, and how consistent are the patterns across time?
So there's individual differences in performance, but once an individual establishes a baseline on the task, it's very stable. And what we do see is generally very orderly changes at the individual level with administration of the dose. It looks like our normal data does, and it generally jives with the self-reported impairment and the time course and magnitude that we see with other computerized tests of performance.
Can you use pupil dilation as a marker proxy for impairment?
We have not seen any relation between impairment and pupil diameter. Eye tracking, there's some signal and some people doing research in that area. We're not doing that currently, but there's some folks that are looking to collaborate with us to build that out.
How did confidence and ability to drive relate to observed change in SDLP in the study?
I can't answer that question. The simulated driving data is still very fresh and very new. We just kind of have those cursory initial graphs mapped out. If you looked at those two slides that I did show, people kind of, I think, overestimated their impairment a little bit. But again, whether they were adjusting their driving because they felt impaired. I know that there's that compensatory driving performance thing that may explain some of it, but we haven't been able to do a deep dive on that data just yet.
On that testing, was there timing in terms of last consumption and when you tested both for blood alcohol and THC?
So the THC and alcohol dosing were sequential, and they were dosed based on known pharmacokinetic, established pharmacokinetic data, so that the peak concentration for both drugs would occur at the same time. So we give the THC first, and then we give alcohol because it's absorbed faster, and the peak concentration, the peak BAC, should be hitting right at around the same time we get peak blood concentrations and peak pharmacodynamic effects of cannabis.
Thank you for the speech. He has a question related to the addictiveness of cannabis and how addictive are cannabis products? How hard is it to quit using cannabis?
So the answer is yes, it can be addictive. And like any other drug, the risk for addiction or the abuse liability is going to be dependent on individual characteristics, doses used, the route of administration here, and the chemical composition of the products. And so as you saw, as I went through the talk, delta-9 THC seems to be the driving force for drug reinforcement, impairment, and abuse liability. And historically, that's really been the driving factor there. We don't see abuse liability of cannabidiol or CBD or CBG or the other minor cannabinoids. I expect you'll have about the same abuse liability for delta-8 THC as delta-9 THC. And as has been shown across many drug classes, and THC is no different, abuse liability is dependent on route of administration. So inhalation has a higher abuse liability than oral ingestion, and oral ingestion has a higher abuse liability than topical administration. So we see very little absorption of cannabinoids through transdermal application. With oral dosing, we do see subjective drug effects. We do see the same kind of reinforcing effects of delta-9 THC as when it's inhaled, but you get a higher abuse liability with inhalation products because you get a more immediate peak drug effect. Neurobiologically, a lot of addiction is triggered by and manifested by dopaminergic changes in the brain and the shorter time course of the drug effects. So you get the more immediate big peak bolus dose and release of dopamine, shorter duration, which means you're using it more frequently and establishing that kind of repetitive drug using behavior. And then it also is going to depend a little bit on the intended purpose of using the product and the dose that's being used. So in cannabis, which is a little bit different from a lot of other drugs of abuse, is that, but not totally different, you have lots of different products, different routes of administration, and different intentions of use. Some people are using it for therapeutic purposes. Some people are using for non-therapeutic purposes. If your intent to use is to get high, you have a higher abuse liability than if your intent to use is to mitigate nausea as a result of chemotherapy. So the amount the dose, and then again, familial history of drug use, individual drug use patterns and history also impact likelihood of problematic drug use.
I think you mentioned this at the beginning of your talk, but the oral dose was edible.
Yes.
Is there a reason you used edible versus sublingual or some other oral ingestion?
Well, we used an edible formulation because that is the most common formulation, common product on the retail market. We were really trying to inform regulatory decision-making and policy with our science. And we're not really on the drug development side. And I haven't seen anything very convincing that sublingual formulations are truly sublingual with cannabinoids and fundamentally different than oral dose formulations. What we see is there's a bigger impact of the actual formulation of the product and the gastric contents of the individual. So we know cannabinoids are absorbed much better when consumed with a high-fat diet or in an oral, an oil formulation, not so much as sublingual versus gastric emptying. I think a lot of sublingual products eventually get swallowed, and that's where a lot of the absorption happens anyway.
Are you planning to increase the numbers of subjects in for stronger results?
So the studies that I showed that are kind of ongoing, the Delta 8 driving study, the pinene study that are ends of 6, 8, 10, we're going to increase to get to 18 to 20 completers for those. But the beauty of within subjects crossover designed experiments is that we don't need much more than about 18 or 20 people to know what the outcome is. And so by eliminating the individual differences and the variance across people and their genetics and all of that kind of stuff, we're able to have a lot of confidence in both pharmacokinetic and pharmacodynamic differences with smaller sample sizes. Now, that being said, we are doing these big observational longitudinal cohort studies with thousands of people. And that'll help us determine, what kinds of individual differences drive different results and reactions and outcomes, particularly for therapeutic indications. So that'll help us determine sex differences, genetic differences, and things like that.
Thank you. We're approaching the top of the hour, so I think this is a good point to sort of close. I want to thank you for the time and for the presentation. This will be available on the ICR website for viewing after this presentation. And just note that Dr. Vandrey has included his email address as well as the Johns Hopkins site. So I thank you again for the time and the presentation. It's a really interesting time in terms of cannabis research, and things may be getting easier in the future, but still great opportunities. So thank you so much.
Yep, my pleasure. Thanks for having me.
Sure.